Good day, everyone, and now we are down to our second to the last topic entitled Parvobacteria and Miscellaneous gram-negative organisms, or the miscellaneous group. So this group includes heterogeneous, small gram-negative fastidious coccobacilli. They are non-motile, non-spore-forming, aerobic, and microaerophilic bacteria. It includes the genus Haemophilus, Bordetella, Brucella, and Francisella. So these organisms need blood or blood products in order for them to grow. As for the learning outcome of this lesson, so at the end of this course or lesson, so students should be able to identify the different species of small pleomorphic gram-negative bacilli, their general properties, virulence factors, and the spectrum of the disease. Second is to explain the pathogenesis of the diseases caused by small pleomorphic gram-negative bacilli, and lastly, explain the process of identifying small pleomorphic gram-negative bacilli in terms of morphology, cultural, growth factor requirements, biochemical, and serological characteristics. Now let's discuss first the genus Haemophilus. Haemophilus species are gram-negative coccobacilli, similar in ultrastructural features to other pathogenic bacilli. They are also pleomorphic. And Haemophilus influenzae, which is a species of our Haemophilus, requires hemin, otherwise known as factor X, and NAD, or also known as your factor V for growth. And other Haemophilus species require only NAD and therefore grow on blood agar. So typeable H. influenzae isolates are classified on the basis of 7 antigenically distinct capsular polysaccharides. So isolates lacking these polysaccharides are called non-typeable. So aside from those, our Haemophilus species are non-motile, non-spore-forming, and they are facultative anaerobes. So most species are oxidase and catalase positive, and they prefer 5 to 10% incubation with your carbon dioxide at 35 to 37 degrees Celsius. Haemophilus species are very prone to drying and chilling, so you must avoid and prevent uh, the organism from the said conditions. So the genus Haemophilus includes a number of species that cause a wide variety of infections but share a common morphology and a requirement for blood-derived factors during growth that has given the genus its name. So Haemophilus influenzae is the major pathogen which can be separated into encapsulated or typeable strains, of which there are 7 types. So A through F, including E. So based on the antigenic structure of the capsular polysaccharide and in unencapsulated or non-typeable strains, as what I have mentioned earlier. So type B, Haemophilus influenzae, is by far the most virulent organism in this group. So commonly causing bloodstream invasion and meningitis in children younger than 2 years. Non-typeable strains are frequent causes of respiratory tract disease in infants, children, and adults. Other Haemophilus species cause disease less frequently. Haemophilus parainfluenzae sometimes causes pneumonia or bacterial endocarditis, and Haemophilus ducreae causes chancroid, and Haemophilus aprophilus is a member of the normal flora of the mouth and occasionally causes bacterial endocarditis. Haemophilus aegyptius, which causes conjunctivitis and Bacillian purpuric fever, and Haemophilus hemolyticus used to be separated on the basis of their ability to agglutinate or lyse red blood cells, but both are now included among the non-typeable Haemophilus influenzae strains. So always remember that most species of our Haemophilus are non-pathogenic except for Haemophilus influenzae, Haemophilus ducreae, and Haemophilus aegypticus. So as what I have mentioned already earlier, that Haemophilus species are gram-negative coccobacilli in that they share common ultrastructural features with other gram-negative bacilli. So the term Haemophilus comes from the Greek word which means blood-loving. So their cell wall contains lipo-oligosaccharide, which resembles the lipopolysaccharide of gram-negative bacilli but has shorter side chains, hence the designation oligosaccharide rather than polysaccharide. Haemophilus species have generally been thought not to make toxins or other extracellular products that account for their ability to produce infection. So these organisms require hemin or factor X and or nicotinamide adenine dinucleotide or DNAD, which is also known as your factor V for growth, whereas NAD is released into the medium by the red blood cells and is available to the bacteria in the blood agar. Hemin is bound to red blood cells and it is not released into the medium unless the cells are broken up as in a chocolate agar. Now let's proceed to the isolation of our Haemophilus species. So the special growth requirements for identification, so as what I have mentioned from the previous slides, So we have the X factor and then the V factor. So X factor is otherwise known as your hemin or hematid. So this is from the iron-containing compounds of our red blood cells. And then V factor is otherwise known as your NAD or your coenzyme 1. So this V factor is produced by some bacteria such as our S. aureus, our S. pneumoniae, and Neisseria species. X factor is a heat stable factor while V factor on the other hand is heat labile. We also have the next test aside from the X and V factor requirement. So we also have the D-ALA test or the delta aminolevulinic acid test, or otherwise known as your porphyrin test, or it is also known as your ALA porphyrin test. So the reagent is our PDAB or paradimethylaminobenzaldehyde, and then the D-ALA test determines the X factor requirement. So in order to detect for the requirement of X factor, so we have to test whether non-hemin requiring species have the enzyme necessary to synthesize heme precursors such as porphyrin and your porphobilinogen from our aminolevulinic acid. So the principle, so strains that lack the enzymes necessary to convert your ALA to protoporphyrin are dependent on the X factor to grow. So our Haemophilus parainfluenzae is ALA positive because it converts your ALA to porphobilinogen and porphyrin to hemin or our X factor. Haemophilus influenzae on the other hand is ALA negative and it needs exogenous X factor. So the positive, while Haemophilus influenzae is positive under UV light, so it produces a red fluorescence at 360 nanometers, which indicates the presence of porphyrin. Next, we also have our culture media. So sheep's blood agar is not recommended for Haemophilus influenzae due to the NAD requirement of the organism. So sheep RBCs releases NADase enzyme except when grown with our Staphylococcus. So the best culture media to be used is our horse or the rabbit blood agar. It's because it has no NADase enzyme. But the most preferable culture media is our cap or the chocolate agar in order to release the X factor. Sometimes we also have our Levinthal agar or it's our Levinthal chocolate agar. So it is just a nutrient agar added with your 5% horse blood with X and V factors. So the encapsulated Haemophilus influenzae on this medium will produce iridescent colonies, and then the non-encapsulated strains of our Haemophilus influenzae are transparent, bluish, and non-iridescent in appearance of their colonies. So on this picture, uh, BAP culture showing Haemophilus influenzae satelliting around Staphylococcus aureus, and this is responsible for the phenomenon of satellitism, in which Haemophilus influenzae forms colonies in the vicinity of our staphylococcal colonies. So satellitism is a distinguishing factor or characteristic of our Haemophilus influenzae. So as you can see on our sheep's blood agar, the Staphylococcus was strict. So unknown is grown with RS aureus, and then Haemophilus species will form small dewdrop colonies, and that's what we call the satellitism. So when grown on our sheep's blood agar, the beta hemolytic strain of our Staphylococcus aureus will release X factor as the red cells are hemolyzed. So as you can see, the colonies of Staphylococcus aureus producing the V factor, and as well as there are small colonies of our Haemophilus influenzae away from the colonies of our Staphylococcus aureus but agar plate, providing the X factor. So that's what is happening here, and that's the phenomenon known as your satellitism. And the next one, we also have your horse blood bacitracin agar. So it contains your beef heart infusion, peptone, yeast, and then 5% defibrinated horse blood. So this one will provide both the X and V factor requirement of our Haemophilus species. And as well as, lastly, it contains 300 milligrams per liter of your bacitracin, and the purpose is to inhibit the normal flora that can be also included in the culture. So the next one is via the use of X and V factor. So this is for the speciation of our Haemophilus species. So the principle, we have to use our Miller-Hinton agar, and then you can also use the XV and then the XV filter paper discs. So are we going to interpret this test or this method? So if there is growth around XV disc, so X and V factors are required, as you can see on the upper right portion of the picture on our MH agar. And then if there is growth around X and XV discs, X factor is required. And then lastly, if there is growth around V and XV discs, V factor is only required. So this table shows the comparison between the different Haemophilus species and their X and B growth factor requirements, and as well as the hemolytic pattern or hemolysis, and lastly, their reaction to our D-ALA test. So kindly familiarize or memorize this table. This also shows the pathogenicity of the various Haemophilus species. So on the left column, we have the various species, and then on the right column, it shows the disease association of these organisms. So please familiarize also this one. Now let's tackle more about the species of our Haemophilus one by one. So let's start with our Haemophilus influenzae. Haemophilus influenzae is also known as your Pfeiffer's bacillus. So as what we have mentioned earlier in the previous slides, that Haemophilus influenzae was divided into encapsulated and non-encapsulated strains. So non-encapsulated strains are part of our upper respiratory tract of the humans. And then our Haemophilus influenzae can cause meningitis commonly in children less than 5 years of age. And they can also cause respiratory tract infection, so both in children and adults. So the general characteristics of our Haemophilus influenzae, so as they are part of the Haemophilus genus, so they are gram-negative coccobacilli. They have smooth drop-like colonies on our chocolate agar plate, and then they need X and D factors on our nutrient agar, and they show satellitism near S. aureus colonies. They also show pleomorphism from our clinical specimens. So Haemophilus influenzae, it is the most pathogenic species of our Haemophilus genus. And then capsule is made of polysaccharides, and it is antigenic. So it increases the pathogenicity of the strains. And then our Haemophilus species will also prevent phagocytosis. And capsulated strains are divided into 6 serotypes. So we have A, B, C, D, E, F. And the most pathogenic type is our type B. And then Haemophilus influenzae invasive infections, mainly type B, include meningitis, epiglottis, cellulitis, arthritis, and our osteomyelitis. Haemophilus influenzae has also various virulence factors, one of which is the capsule. So capsule has the most significant role in the virulence, so as it will help the organism to evade phagocytosis. And then IgA1 protease, so these are proteolytic enzymes, which are produced by our pathogenic bacteria that will infect and colonize mucosal surfaces. And then fimbriae, these are long filamentous polymeric protein structures located at the surface of our bacteria, and then they will enable the bacteria to bind to specific receptor structures and thereby colonize specific surfaces. So that's why they will adhere. And then our lipopolysaccharides, these are the major outer surface membrane components, which is present in almost all of our gram-negative bacteria, and extremely strong stimulators of innate or natural immunity in diverse eukaryotic species ranging from insects to humans. So as I recall, encapsulated strains are pathogenic, and then main cause of meningitis in children less than 5 years old, this was already mentioned from the previous slide, associated with respiratory conditions including epiglottitis, and then vaccines was available. So these vaccines were prepared from the capsular material of our Haemophilus influenzae in order to destroy the capsule. And now let's proceed to the laboratory diagnosis of our Haemophilus influenzae. So we have a lot of specimens. So we have CSF, blood, sputum, swabs, and synovial fluid. So if you're suspecting for meningitis on our patients, we can collect CSF and as well as blood. So blood can also be used for bacteremia, etc. Sputum for pneumonia, swab for cellulitis, and synovial fluid, of course, for our arthritis. So on a smear, so you can observe pus cells plus the pleomorphic gram-negative coccobacilli. And then if you're going to use the CSF as the specimen, so you're going to culture it on our chocolate agar plate, and then you're going to detect the presence of our capsular antigens by agglutination. Now let's proceed to our Haemophilus aegypticus. So Haemophilus aegypticus is otherwise known as our Kochwik's bacillus, and then it causes pink eye conjunctivitis, as well as the Brazilian purpuric fever. Kochwik's bacillus is also otherwise known as your Haemophilus influenzae biotype 3. So our pink eye conjunctivitis is a contagious disease which is spread through sharing of handkerchief and towels. Next, we also have our Haemophilus ducriae. It is the causative agent of our soft chancroid or chancre. So how do we define this one? So it is a venereal disease characterized by painful ulcers in the genitalia, as what you can see in the picture. And please remember that Haemophilus ducriae is the smallest pathogenic bacilli. So in gram stain, they resemble schools of fish. And then our Haemophilus ducriae needs X but not V factor. So they are very fastidious, And you can use freshly clotted rabbit, sheep, or human blood for culture. And then, as what I have mentioned in the previous discussion, you can also use your chocolate agar plate with 1% isovitalics or gonococcus agar with 1 to 2% mercury, and then 5% bovine fetal serum and 3 micrograms per ml of arbamycin. So direct examination of the genital ulcer, so you can observe, of course, by the help of our gram staining, the school of fish. So this is what it looks like under the microscope. So the schools of fish. So this is comparable to the appearance of our Haemophilus ducreae in a gram stain. So we also have our other Haemophilus species. So next we have our Haemophilus aprophilus and our Haemophilus paraprophilus. So they are only one species, so coming from our aggregate Bacter aprophilus. So they are oral and respiratory tract flora of our body. And then if they will cause infection, so they can cause endocarditis and pneumonia. We also have our Haemophilus hemolyticus. They require X and V factor, and then they are hemolytic organisms. So sometimes they are mistaken as your group A streptococcus. Next, we have your Haemophilus parainfluenzae. So they are part of the human respiratory tract flora, and then they need V factor only. So they resemble Haemophilus influenzae, and parainfluenzae also has the same disease association with Aprophilus and Paraprophilus. So next we have Haemophilus suis. So they resemble also Haemophilus influenzae, but Haemophilus suis causes disease in hogs. And lastly, we have your Haemophilus hemoglobinophilus. So they need X factor but not B factor, and this is found only in dogs but not in humans. Since we are done tackling with the genus Haemophilus, now let's proceed to the genus Bordetella. So all are non-motile except our Bordetella bronchioseptica, and then the ideal specimen is our nasopharyngeal swab. So Bordetella, they are aerobic and oxidative obligate parasites in animals and humans. So Bordetella pertussis, so this is the most common one or the notorious one. So this causes whooping cough. And then our Bordetella parapertussis, so it causes pertussis-like syndrome, which is the milder form of our whooping cough. And our Bordetella bronchioseptica, or otherwise known as your Bordetella bronchicanis, so this causes kennel cough or snuffles, no? So and it causes pertussis-like illness when it infects humans because Bordetella bronchioseptica uh, causes zoonotic infections or in animal diseases. And we also have your Bordetella avium. So it is found in our turkey, coryza. So there was no known human infection. And then we also have our newer isolates. So we have your Bordetella hinzii. We also have your Bordetella holmesii and your Bordetella trematum. So Bordetella pertussis, parapertussis, and bronchioseptica, they are closely related with 72 to 94% of our DNA homology with little differences in their multilocus enzyme analysis. Now let's discuss them one by one. So let's start with Bordetella pertussis. As what I have mentioned earlier, that our Bordetella pertussis is the agent of our whooping cough. So they are small, encapsulated gram-negative coccobacilli, and they are non-motile. So using toluidine blue, we can see bipolar mutachromatic granules under the microscope. And then the culture media that is most preferable is our charcoal containing medium. So of course, the incubation temperature is at body temperature, and it usually runs from an incubation period of 3 to 7 days in a moist environment. So they are strict aerobes, and they form acid but not gas from glucose and lactose, and they are oxidase and catalase positive, while on some biochemical tests such as nitrate, citrate, and urea, they are negative. Our whooping cough has 3 stages. So the first stage of our whooping cough is the catarrhal stage. So it is a mild cold-like infection. So it is highly infectious but not very ill. So the second one is our paroxysmal phase or the paroxysmal stage. So in here you can see copious mucus, and then it will cause narrowed epiglottis, and then that will be the reason, no, the narrowing of your epiglottis, that you will whoop, and then the cough will interfere with your swallowing. So the swallowing of the mucus will also cause vomiting, and then this vomiting can result to dehydration. And then hypoxia, if there are prolonged attacks of whooping cough, it can lead to seizure, and then hypoxic encephalopathy or even coma. So the last stage is our convalescence or the convalescent stage. So this is the recovery phase. So the cough episodes will slowly decrease, and then there will be a gradual recovery over 3 to 16 weeks. And then, if not treated, so our whooping cough have secondary complications. So we have pneumonia, otitis media, rectal prolapse, and even meningoencephalitis. Our Bordetella pertussis is part of the vaccine DPT. So this is a trivalent vaccine. So it stands for diphtheria, pertussis, and tetanus. So this is a vaccine given to our infants. So the virulence factor of our Bordetella pertussis is our pertussis toxin. So the functions are the following. So it increases histamine release and it will enhance inflammatory response, and then it contributes to bacterial binding to our ciliated epithelial cells. And lastly, it will prevent the deactivation of our adenylate cyclase. So aside from our pertussis toxin, Bordetella pertussis also possess heat stable endotoxin or our LPS and our exotoxins. So exotoxins, we have the filamentous hemagglutinin. So that's the responsible for attachment to our ciliated cells. So and then adenylate cyclase toxin. So this is an antiphagocyte. We also have the tracheal cytotoxin, which is ciliostasis and damage to our ciliated epithelia. And lastly, the dermonecrotic toxin, which causes tissue destruction. Now let's proceed to the isolation of our Bordetella pertussis. So they require isolation media, and then the agar that you can use is our boarded gengo agar or the BG agar. So what you can observe are small colonies in 7 days. So they are capnophilic and they should be incubated at a temperature of 35 to 37 degrees Celsius. The BG agar contains potato, glycerol, and blood, and colonies resembles mercury-like droplets as you can see on the picture. So patients with suspected whooping cough simply cough into our BG plate or the so-called cough plate. We also have the Regan low medium aside from our BG agar. So the Regan low medium contains the horse blood, charcoal, cephalexin, and our amphotericin B. So this is preferred by most of our microbiologists, the Regan low medium. As for the specimen of our Bordetella pertussis, you can use a saline nasal wash or of course, the cough plate nasopharyngeal swabs. So it should be placed in a Dacron or Ryan swabs. And you can perform direct FAT. So this direct FAT is very useful in identifying Bordetella pertussis after culture on a solid media. So if you are going to perform FAT or DFA, so you can have your slide agglutination with a specific anti-serum. And of course, our most sensitive and most reliable method, you can perform PCR. So this is the most sensitive method to diagnose Bordetella pertussis. Bordetella pertussis, as what I have already mentioned from the previous slide, that it can be used in a vaccine. So the trivalent vaccine, DPT, which stands for diphtheria, pertussis, and tetanus. So 3 injections of pertussis vaccine is given to our infants. So first year of life, you can also have a booster of 5 doses. So it will be given intramuscularly as part of the triple vaccine. Next, we have the table for the different Bordetella species. So our Bordetella pertussis, they can show brown pigment on our BG agar, they are non-motile, and they can cause pertussis-like syndrome. While Bordetella bronchioseptica, they have a Petricus flagella. They can cause septicemia and meningitis, and they are rapid urease producers. We are done with the Brucellae. We are done with the Bordetella. So now let's proceed to the next organism under your Parvobacteria, which is the Brucellae. So our Brucellae are obligate intracellular parasites both in animals and humans, but they are inactive metabolically. So our Brucellae, they are short gram-negative cocobacilli, and then they are aerobic and non-motile and non-spore forming. So on our gram staining, in order to stain our Brucellae, you must replace safranin with carbolfuchsin. So our Brucellae is the agent of brucellosis or the undulant or Malta fever. So you can acquire this one via ingestion of our contaminated animal products like meat and milk. And the direct contact for this kind of organism are our farmers. So next, for the blood culture, uh, you can have a blood culture if there there is already febrile illness, usually 2 weeks. And then, so it will be negative after acute symptoms will subside on our patient. So we can also have, you can also use Casanilla de Battles, the biphasic system, the febrile agglutinin, which is the gold standard. So our biphasic culture media, so or the Castañeda bottle, it requires 5 to 10% carbon dioxide for 30 days. Okay, so while our Castañeda bottle, or Castañeda medium, you can also use bone marrow and blood, so in which brucella are most isolated. So what you can observe is that you can find a tiny gram-negative cucobacilli, and when you test for oxidase and catalase, so they are positive. And then uh, you can also perform this one on a reference lab, so via hydrogen sulfide, dye inhibition, and agglutination sera. So on a culture-enriched media, so the appearance of our brucella species are small, convex, smooth colonies, and they require amino acids, vitamin salts, and glucose. All of our brucellae or our brucella species can grow in ambient air except for our brucella abortus, which requires 5 to 10% of carbon dioxide. They are catalase, oxidase, and nitrate reduction positive, and some of them are hydrogen sulfide producers. So this is the table for the differentiation of the various species of our brucellae. So Brucella militensis, this is the only species by DNA studies, and it has high concentration of our M antigen. So Brucella militensis is known to cause Malta fever, or the brucellosis, or the undulant fever. And then Brucella suis, so it will only cause you a mild disease without suppurative complications and non-cacheating granulomas of our reticuloendothelial system. Brucella abortus, on the other hand, is otherwise known as Bangs bacillus or Bang bacillus. So abortion in cattle. So this is the disease association of our Brucella abortus. Thus the name abortus. And then it contains high concentration of A antigen. And lastly, our Brucella canis. So it can cause chronic and suppurative lesions with cacheating granulomas. So you should be very careful when handling or processing specimens suspected with the organism Brucella, because they are really hazardous organisms. So that's why you are going to perform this one in a reference public health laboratory with biologic safety cabinets. Now let's proceed to the next organism, which is Francisella. So the most common organism under the genus Francisella, or the species, is our Francisella tularensis. And this is the agent of our tularemia. Tularemia, it is a disease of rodents, primarily in our rabbits. How do we get this one? So if you have direct contact with the blood or through animal scratch, or you have been bitten, so it has invasive properties that allows to penetrate the skin. So vectors for our Francisella tularensis, you have deer flies and ticks, and then the special medium is our cysteine glucose blood agar. And of course, the same with our Brucella, extreme caution should be taken when handling the organism. Our Francisella tularensis have subspecies such as Francisella tularensis subspecies tularensis, Francisella tularensis subspecies hulartica, Francisella tularensis subspecies mediasciatica, and Francisella tularensis are non-spore forming and they are non-motile. And of course, they are encapsulated and they are biochemically inactive. So Francisella tularensis was also used as a biological weapon. So they are extremely invasive and infectious because they can be transmitted via aerosol. So it was classified under a biohazard 3 safety measures if you are going to process Francisella tularensis. So aside from the vector mode of transmission, you can also, if you have ingested contaminated food and water, you can get infected by this one. So the tularemia is somehow similar to plague, but it has a lymph node involvement. Now let's proceed to the lab diagnosis of our Francisella. So the specimen is the ulcer scraping, we have the lymph node biopsy, bone marrow, and then sputum and serum. So under the microscope, they are tiny gram-negative cocobacillus, and on a culture, they are slow growers, so they should be grown on a special media for at least 2 to 3 days. And then you can use BCYE or glucose-cysteine blood agar with thiamine and a supplemented CYA. So on serologic test, you can use FAT or the ELISA. And please take note that smears and culture are not contributory and diagnosis rests on the serologic studies. Now let's proceed to the miscellaneous group or the other fastidious bacilli, gram-negative bacilli. So we have our Campylobacter. So this is the most recognized antecedent cause of our Guillain-Barré syndrome. So they are microaerophilic, which means they require 85% nitrogen, 5% oxygen, and 10% carbon dioxide. And you can be infected by this one via fecal-oral route. Clinical signs of Campylobacter infection, so you can have fever, abdominal pain, and diarrhea, and stool under the microscope will contain segmented neutrophil and our red blood cells. So this is only in cases of severe gastroenteritis. And we also have the Campylobacter fetus, subspecies fetus. It's the agent of infective abortion in cattle and sheep. So we have the culture media. So the selective media, we have Campylobacter blood agar and your Campylobacter agar sciro. So the differences is on the contents of our various antibiotics. So on the left side of the photo, that is an electron micrograph of our Campylobacter jejuni. And on the right side of the photo, that's a seagull. And the seagull's wings is said to be comparable to the morphology of our Campylobacter jejuni. So they also show darting motility. So on a stained smear, so they are S-shaped or wings of seagull. So the optimum growth for our Campylobacter jejuni or jejuni, which is the most common cause of bacterial gastroenteritis, is at 42 degrees Celsius. Next, we also have our Helicobacter. So Sir J. Robin Warren and Sir Barry Marshall swallowed a good-sized inoculum in order to prove that Helicobacter pylori exists. So they won a Nobel Prize on this one. And then the result is a short-term case of gastritis. The most common isolate is our Helicobacter pylori. It is the agent of peptic and duodenal ulcers. The natural habitat is our human stomach, and then Helicobacter pylori are motile with 4 to 6 polar flagella, and you can use urea breath test for this organism. So if it is positive, it is a strong urease producer. Histologic staining and culture biopsies obtained from the stomach or duodenum is the recommended method or specimen for our identification. But you can also use Campis, BAP, and Scaros as well as the same with your Campylobacter species. So in order to differentiate the two organisms, so we can use urease test, hybrid hydrolysis test, and growth at 42 degrees Celsius. The next one is our Legionella. So the Legionella are catalase positive. And then we have various species of our Legionella. So we have Legionella pneumophila. It is oxidase positive and it hydrolyzes hyperate. So most Legionella produce gelatinase and lactamase except for our Legionella mycdadi. So Legionella mycdadi is the causative agent of Pittsburgh pneumonia, while Legionella pneumophila is the agent of Legionnaire's disease or Pontiac fever. We also have your Legionella bozzemani I. It is the weakest agent of pneumonia. So all of them require cysteine for growth. And then as for the specimen of this organisms, we can use bronchial washings, pleural fluid, lung biopsy specimens, or blood, and then sputum is not preferred. So for the culture, we have buffered charcoal yeast extract with alpha ketoglutarate, or the buffered charcoal yeast extract is the best medium for our Legionella species. So you can observe pinpoint ground glass appearance, round or flat, colorless or varying color, translucent or speckled, average of 3 days. If you are going to use blood agar and McConkey agar, then there will be no growth on this one. Now let's discuss first our Legionella pneumophila. So this is the major cause of diseases in humans, and then it was widely publicized outbreak of pneumonia in Philadelphia. So it was found in the American Legion Convention delegates. That's why it was known as your Legionnaire's disease. So patients infected with this one will have flu-like symptoms, then dry cough, diarrhea, pneumonia, liver, and renal involvement. So how you can get infected by this organism? So ingestion or inhalation of aerosols from contaminated cooling towers. And then Pontiac fever, it's somehow a flu-like illness. And then the virulence are the intracellular survival within the macrophages of our organism. So you can use charcoal as a detoxifying agent. And then this can be made by selective addition of our cefamandole, anisomycin, and or polymycin B. So bodies of water, lakes, streams, rivers, especially thermally heated bodies of water and soil. So these are the natural habitats of our Legionella pneumophila. And then the virulence factors of our Legionella pneumophila. So we have inhibition of the phagosome-lysosome fusion, which allows for the intracellular growth of our bacteria. They also have endotoxin. They also inhibit generation of bactericidal substances in phagocytic cells. And they also have hemolysin and collagenase. So for the clinical significance of our Legionella pneumophila, it was already mentioned earlier. So it can cause acute pneumonia or Legionnaire's disease. And then airborne transmission with an incubation of 2 to 10 days is the mode of transmission. And then the symptoms, if you have this one, you can have fever, chills, myalgia, headache, dry cough, vomiting, diarrhea, and abdominal chest pain. So hospital is usually required in 3 to 5 days. So without antibiotics, the fatality rate of this organism is as high as 15%, and then the disease occurs more in males over 60 years of age and in the immunocompromised. The reservoir of infection is often in the cooling towers of air-conditioned systems and in hot water lines, as well as in soil and water. So the Pontiac fever, so it's an acute self-limited febrile illness with an incubation of 24 to 36 hours. And then the symptoms include high fever, chills, myalgia, and headache, which lasts 2 to 5 days. And then the treatment or the antimicrobial susceptibility drug for this one is our erythromycin, rifampin, or tetracycline. Next, let's proceed to our Bartonella species. So we have Bartonella bacilliformis and our Bartonella hinseleyi and quintana. So all of these Bartonella species are gram-negative rods. They are pleomorphic, they are slow-growing, and they can be seen in infected tissues with warthin, starry, silver impregnation stain. So Bartonella bacilliformis, is the causative agent of our Oroya fever. Oroya fever is the initial stage of the disease, and then it can cause a serious infectious anemia. And then the eruptive stage of our Bartonella bacilliformis is our Verruga peruana. So usually it erupts at a period of 1 year, and it will cause you vascular skin lesions or crops. So it is transmitted by our sandflies such as the Orlotzomia, and then this organism or these sandflies are limited to Peru, Colombia, and Ecuador. While Bacillus cencilei and Bartonella quintana is the causative agent of our cat scratch disease. So it can cause fever, lymphadenopathy. So how are you going to acquire this cat scratch disease? So by contact with a cat, so via its scratch, via licking, or you have been bitten or flea bite of the cat. And then it can also cause bacillary angiomatosis, which is an enlarging red cranberry-like purple, often with surrounding scale and erythema, which is found on the picture. Next, we also have our Gardnerella vaginalis. Gardnerella vaginalis is the causative agent of our bacterial vaginosis. Bacterial vaginosis is a premature rupture of the membranes and preterm labor and birth. So the odor of this organism, if present in a vaginal discharge, it will have a fishy odor, and then it will have no inflammatory cells present. And then the pH is over 4.5 to the normal pH of the vaginal discharge is less than 4.5. So in a direct wet mount, you can observe clue cells. So clue cells are just desquamated epithelial cells with attached organisms. Gardnerella vaginalis is treated with metronidazole or non-selective vaginalis agar, which contains human blood, and it produces diffuse beta hemolytic colonies, and they can only grow in 5 to 10% carbon dioxide for 48 hours or in an anaerobic condition. So for the diagnosis, they are gram variable, so they can be gram positive or gram negative. They are catalase negative, and they can grow on our CBA. And then you can observe beta hemolysis on our vaginalis agar, and of course the observation of our clue cells on a direct wet mount. Clinical significance of our Gardnerella vaginalis, they can be part of the normal flora of the vagina, and they can act synergistically with anaerobic bacteria to cause a bacterial vaginosis characterized by a thin, milky vaginal discharge with a fishy odor upon the addition of our potassium hydroxide. And this organism may be involved in maternal and neonatal septicemia. The next organism we have is our Capnocytophaga. So they are capnophilic and then falciform in appearance, or they are filamentous bacilli. So the most distinguishing characteristic or striking characteristic of our Capnocytophaga is the gliding motility. Next, we have our Calimactobacterium granulomatis. Calimactobacterium granulomatis, it's the agent of granuloma inguinale or dunovanosis. So our Calimactobacterium granulomatis is somehow related to our Klebsiella. So our Whipple's disease is also caused by our Calimactobacterium granulomatis. So you can have fever on this one, abdominal pain, diarrhea, weight loss, and migratory polyarthralgia. Whipple's disease is also caused by the organism Trophirema whipplei or Whipple eye. So aside from Calimactobacterium granulomatis, we have your Chromobacterium violaceum. So they are motile, facultative, anaerobic, and then they are oxidase positive, and they produces the pigment violacin, which produces a violet or a purple color. And then we also have your Cardiobacterium. They are normal upper respiratory tract flora. They are non-motile, and they are oxidase positive and catalase negative, and as well as indole positive. And we also have your Plesiomonas shigelloides. So this is an important organism in Japan as a cause of gastritis. Our Plesiomonas shigelloides can grow on our BAP, MAC, and also on our EMB. Next, we have your Echinella corrodens. So they are capnophilic rod, and then they are part of the gingival and bowel flora. So the most distinguishing feature or characteristic of our Echinella corrodens in an agar medium is the corroding or pitting of the agar. The next organism that we have is our Streptobacillus. So the most common isolate is our Streptobacillus moniliformis, and it is the agent of our rat bite fever. And then it will have a septic relapsing fever, blotchy, and petechial rashes if you will have rat bite fever. And then you will also have painful polyarthritis. Our Streptobacillus moniliformis also causes Haverhill fever, in which you can get this one if you will ingest contaminated milk. So Streptobacillus moniliformis are aerobic, they are pleomorphic gram-negative rods, and they have irregular chains. So in a culture media, you can observe L forms of our Streptobacillus moniliformis. So they are best grown at 37 degrees Celsius and they cease to grow at 22 degrees Celsius. So the media with serum, protein, egg yolk, or starch is preferred for our Streptobacillus moniliformis. They are also biochemically inert. You can, for serologic test, you can use serum agglutination test, and then the treatment of choice for Streptobacillus moniliformis is our penicillin. We also have your Spirulum minor, so it will also cause rat bite fever but is different in clinical appearance, the Sudoku fever. So we also have your Yersinia. So we have Yersinia pestis. Yersinia pestis, they are gram-negative bacteria or bacilli that inhibit striking bipolar staining, and then they are non-motile, and then they are facultative and aerobes. So Yersinia pestis grow rapidly in media with blood or tissue fluids, and then the mode of transmission for our Yersinia is via flea bites or inhalation. So the flea, the scientific name of the flea is Synopsilla chiopis. So it causes black death or it's the agent of plague. And then symptoms, so you will have fever and painful lymphadenopathy with enlarged tender nodules or the so-called bobos in your groin or in your axilla. So you will also experience vomiting and diarrhea, and then early septicemia and sepsis. You can also experience disseminated intravascular coagulation, so which will result to hypotension, altered mental status, renal and cardiac failure. And aside from that, Yersinia pestis can also cause pneumonia and meningitis. So for the laboratory diagnosis, you can have blood sputum and the CSF specimens, Wieson stain that will exhibit the bipolar appearance of our Yersinia, and then streptomycin is the drug of choice. So Yersinia pestis will multiply intravascularly, so that's why it can be seen in our blood smears. And then on a CIN agar, so you can observe bullseye colonies with red center for our Yersinia enterocolitica. Yersinia enterocolitica also is a blood bag contaminant. And now we are down to our last organism, so under Pasteurella. So we have Pasteurella or Pasteurella multocida. This is the major pathogen of this genus. So this is zoonotic bacteria, and Pasteurella multocida is the agent of shipping fever in cattle and animal bite or scratch wounds. So the general characteristics of our Pasteurella multocida, they are gram-negative pleomorphic coccobacilli. They are also exhibiting bipolar staining. They are non-motile. They are facultative and aerobe. They can grow on BAP and CAP, and they are oxidase and catalase positive. So the disease association of Pasteurella multocida is our pasteurellosis. So they will not grow on your MAC and EMB, so only on our BAP and CAP. Pasteurella multocida is susceptible to most antibiotics. And then penicillin G is the drug of choice for our Pasteurella multocida. Tetracycline and fluoroquinolones are the alternative drugs of our Pasteurella multocida. And that ends our discussion or lesson under Parvobacteria and Miscellaneous group. I hope that you have learned something, everyone. Thank you and have a good day.